So welcome guys once again, and today our topic around the virology short notes. And all you know that we are going through a series of Aiims CRE. It's not only for Aiims CRE, all other exam can include in this. We are just making short notes, and today we are discussing about the virology, right? So first of all, our is introduction to virology. What is virology? All the thing is similar. Logy means study of the thing and viro means virus. So virology is a study of viruses. क्या है ये? Infectious agent smaller than bacteria. They are the infectious agent and which are the smaller than the bacteria. They can replicate only inside the host cell. One of the important thing is that when we are culturing the bacteria, so we can cultivate in the artificial medium like nutrient agar, blood culture in this. But for the virology, we have to must require a host cell or a living cell so that the virus can replicate on it. So this is a very important thing about the viruses. They're acellular, so single cell, non-living, outside the host. One important thing about the this viruses are they are the link between the. So one important thing is that these viruses are linked between the living and non-living being. The viruses are non-living when they are outside the human body or any apart from the host. But when they are inside the host, so they act as a living being. So this is a very typical feature of these viruses and uniqueness of the nature, right? So they are composed of nucleic acid. Everything have a certain nucleic acid. But in case of viruses, either it is DNA or RNA. This is very important. Viruses never contain both DNA and RNA. Exam point of view से बहुत important point है ये. Then there is a protein coat inside this DNA or RNA. There is a protein coat which is called as a capsid, right? Some viruses have an envelope. Outside this capsid, there is a layer, right, made of lipid membrane. This is capsid is made of a protein. Very important. Capsid is made up of protein coat, while lipid membrane is made up of envelope is made up of lipid membrane, right? And these are lipid derived from the host cell, right? So if we make a structure, suppose this is our DNA or RNA, outside there is a capsid, and outside this there is a envelope which is made up of lipid, right? Sometimes if this envelope is not, so we see only the up to the this capsid area, right? We can't see the envelope. So based on this, viruses are divided into enveloped and non-enveloped, right? Obligate intracellular parasite, I already told you, they are act as a parasite or act as a living being when they are inside the cell. So obligate intracellular for their living being. Require host cell to replicate. Then infect all life form, human and they infect all forms. तो human तो animal हो, plant, fungi, bacteria. बैक्टीरिया बैक्टीरियोफेज हमने पिछली वीडियो में आई थिंक पढ़ा भी इस चीज को, बैक्टीरियोफेज को, राइट? बैक्टीरिया के ऊपर भी अटैक करेगा एंड देन ट्रांसफर करता है जीन को इधर उधर. सो इट अफेक्ट द ऑल फॉर्म, राइट? नाउ वी गो फॉर द स्ट्रक्चर ऑफ द वायरसेस. आई ऑलरेडी टोल्ड यू दिस वन, राइट? इट्स क्वाइट सिमिलर. सो फर्स्ट ऑफ ऑल वी हैव अ कैप्सिड, protein coat protecting the genetic material. So you have I already told in the bacteriology video, we have a spore, so they also act as to protect the bacteria, but here the capsid protect the genetic material which is the DNA or RNA, right? And some of contain the outer layer also which is made up of lipid layer in some viruses like HIV and influenza, spikes, and some of the viruses there is a spikes, right? This type of spikes like you can see in the coronaviruses. So they are a glycoprotein nature, right? Made up of carbohydrate protein mixture, help in attachment to the cell. So they generally help attachment for the cell. In case of bacteria, fimbriae act as a attachment, right? Genome. So genome is a genetic material which may be either DNA or RNA, never both. Very important, it can be never both. And based on this structure, we have a icosahedral, helical and complex shape. Icosahedral, which contain a 20 sides, I think faces 20. So it is very unique type of structure. I will pick up a photo if I get. Then we have a helical also, and this is helical type complex is kind of bacteriophage, right? It contain like a bacteriophage, right? Now classification of viruses. So based on nucleic acid, we have a DNA virus or RNA viruses. This is very important and a basic classification. We have either DNA virus or RNA virus. DNA viruses are the double standard. DNA viruses are double standard viruses except parvovirus. Parvo is single standard DNA virus. This exception is very, very important for exam point of view. In case of RNA virus, all the RNA are single standard except reovirus. So it is very important, right? Because the exception list may come in the exam. Based on presence of envelope, some bacteria have envelope and some does not. So envelope bacteria are HIV, influenza and herpes. In case of naked viruses, we have adeno, norvo and papillomavirus, right? So this is very, very important also. Another we have a based on replication site, where they are replicated. Like in case of DNA virus, they replicate in the nucleus because most of the DNA part present inside the nucleus. But so all the DNA virus replicate inside the nucleus except pox virus, they are replicate in the cytoplasm. In case of RNA virus, RNA spread all over the cytoplasm or also in the nucleic acid. Mainly the RNA viruses are replicated in the cytoplasm, but exception are always there. Influenza and retrovirus, they can replicate inside the nucleus. Now another is based on shape. So icosahedral, example are adenovirus and herpes virus. Helical shape virus, influenza and rabies. Complex virus are parvo and bacteriophage. So this is very important. You can understand, right? Then medical importance virus and disease. So there are so many viruses, but we have arranged some of the important viruses, what they cause disease and their key feature, right? Herpes virus, herpes simplex virus NN2, they are sexually transmitted virus. So cold sores may be cause and genital herpes, very, very important, right? It causes genital herpes and where they mainly attack latency in the neuron. So nervous system may be weakened due to this. Then we have varicella zoster virus, disease, chickenpox and shingles. Where is they mainly what cause? Dormant and dorsal root of ganglia. So it is also part of our neuron system. Then Epstein-Barr virus, infectious molecules, which is very important, and this is a question. They can also come in the hematology point of view, right? Infectious molecules caused by EBV virus, Epstein-Barr virus, linked to the Burkitt lymphoma. I already told you the question is linked to the also hematology. In case of lymphoma and especially Burkitt lymphoma, we see the infection nucleus which is caused by EMB, Epstein-Barr virus. Another we have a cytomegalovirus, congenital infection and retinitis in case of eyes. So what we see the owl eyes inclusion, right, in the microscope. Then we have a hepatitis A, B, C, D, E, cause disease hepatitis, HIV A and HIV. So these are both A and E spread through the fecal oral route, while B and C are from bloodborne disease, right? We have an influenza virus, commonly called as flu virus, segmented RNA viruses, antigenic shift drift. So this is not very important, but influenza virus cause flu, right? Then we have rabies virus. It is due to the dog bite, all you know, and the disease cause rabies. Very, very important. The virus structure is bullet shape. The virus structure is bullet shape, right? So this is very important. And we see the Negri body neuron, very, very important point. मतलब अगर मैं बोलूँ कि 100 में से 100 पॉइंट इस पॉइंट को मिलने चाहिए. Negri body seen in the neuron in case of rabies. Another we have HIV, human immunodeficiency virus, cause AIDS, target CD4 and T cell, and it is also known as retrovirus. ठीक है, मेडिकल टर्म में कई बार HIV पॉजिटिव नहीं लिखते, रेट्रो पॉजिटिव लिखते, तो इट मीन पेशेंट इज पॉजिटिव फॉर द HIV. दिस इज ऑल्सो इम्पोर्टेंट पॉइंट. CD4 को टारगेट करता है. सो HIV के केस में हमें CD4 काउंट करते हैं, अब वो लो आएगा, राइट. अनदर वी हैव अ ह्यूमन पेप्लोमा वायरस, कॉज वार्ट्स एंड सर्विकल कैंसर इन केस ऑफ फीमेल, वेरी इंपॉर्टेंट. ह्यूमन पेप्लोमा वायरस 16 एंड 18 आर द अपने CD मार्कर, राइट. इसके लिए हाई रिस्क स्ट्रेन है हमारी ये 16 और 18. सॉरी 16-18 CD मार्कर नहीं है, ये हमारे स्ट्रेन है, मतलब कि स्पीसीज कह सकते हैं इसकी, जो कि बहुत इन्फेक्शियस है। रोटा वायरस डायरिया करता है चिल्ड्रन में, दिस इज ऑल्सो वेरी वेरी इंपॉर्टेंट पॉइंट कि रोटा वायरस डायरिया करता है चिल्ड्रन में। और व्हील शेप वायरस होता है, इस जैसे गाड़ी का अपना व्हील होता है वैसी शेप में इसका वायरस होता है। मेजर कोज गैस्ट्रोएंटाइटिस। डेंगू वायरस एडी डेंगू फीवर से कॉज होता है ये, ब्रेक बोन फीवर। तो इसमें हड्डियां वगैरह सब दर्द होती हैं। अभी जब मौसम आएगा भी डेंगू तो आपको देखिएगा पूरा पूरा तोड़ देता है हड्डियों को पूरी है, इसीलिए इसको ब्रेक बोन वायरस कहते हैं। दूसरा एडिस एजिप्टिस, इसका जो है मॉस्किटो ट्रांसमिशन के लिए काम करता है। देन वी हैव अ कोरोना वायरस SARS-CoV-2, देन MERS SARS, तो ये क्या है, COVID-19 है। देन स्पाइक्स प्रोटीन फॉर द एंट्री, वही जो मैंने बताया कि अटैचमेंट के लिए दे हैव अ spikes, right? Now another point is viral replication cycle. So how a virus replicate? So this is our cell, take an example. First of all attach होगा वो, ठीक है, attach किससे होगा, they have a spikes or some also with the help of receptor also, right? So in case of first point attachment, virus bind to the host cell receptor. So they bind to the host cell receptor. Penetration, उसके बाद penetrate करेगा inside the cell. सो एंट्री विया एंडोसाइटोसिस और मेम्ब्रेन फ्यूजन, या तो इसके जो सेल वॉल है ये से मिक्स हो जाएगी, या दूसरा क्या एंडोसाइटोसिस, अंदर घुस जाएगा पूरा का पूरा वायरस। सो मे बी दे एंटर्ड इन दिस पॉइंट। अनदर पॉइंट वी हैव अ अनकोटिंग। नाउ जो इसके अंदर जेनेटिक मटेरियल होगा हमने डिस्कस किया है यहाँ पे, इस पॉइंट में हमने डिस्कस किया कि क्या क्या होता है पॉइंट इसमें। सो एट दिस पॉइंट दे हैव अ जेनेटिक मटेरियल कैप्सिड ऑल्सो, ऑल द थिंग। सो अनकोटिंग होगी, आउटर लेयर में भी एनवलप की तो वो हट जाएगी, ठीक है, या फिर कैप्सिड हट जाएगा। रिलीज ऑफ वायरल जीनोम, मेनली जो अंदर का ये है हमारा DNA और RNA, ये रिलीज होगा। देन रेप्लिकेशन, अब जो भी है DNA या RNA ये रेप्लिकेट होगा। अब इन केस ऑफ DNA वायरस इट विल रेप्लिकेट इन द न्यूक्लियस, इन केस ऑफ RNA वायरस वो साइटोप्लाज्म में ही रेप्लिकेट होना स्टार्ट हो जाएगा। ये सेल का हमारा समझो कि वो है न्यूक्लियस। तो यहाँ पे ये बहुत सारे वायरस के पार्ट बनना स्टार्ट हो जाएंगे। प्रोटीन ये जेनेटिक मटेरियल मल्टीप्लाई होगा, देन कहीं पे हमारा कैप्सिड के लिए प्रोटीन सिंथेसिस होगी, देन लिपिड के लेयर से लेके एनवलप बनेंगे। सो ऐसे होगा। और फिर होता है नेक्स्ट स्टेप असेंबली, तो इन सब को जोड़ के एक नया वायरस बनाया जाएगा, ठीक है, असेंबली करेंगे। देन रिलीज, फिर जब ये पूरा एक वायरस जो एंटर हुआ ये अपने जैसे बहुत सारे वायरस कर लेगा, ठीक है। The virus when enter it single and it make their progeny in multiple number. Finally they release, ठीक है। Virus are live by budding. So budding yeast के केस में होती है कि यहाँ से ये निकलेंगे धीरे धीरे। In case of antiviral or lysine naked virus, when the virus are enveloped, so at that case they are released by budding system. But in case of naked viruses, when there is no envelope, so they are released by lysis. So they have so many number. In case of naked virus, they will flooded with the baby virus, and after that the cell cannot bear the load and it finally break down, and in this case it called lysis, and they come out of this viruses. So these are the very very important case of application. And if you want a complete video, so already we have in our channel, so you can see the deep and detailed thing about that. Now laboratory diagnosis virus. So हम लेबोरेटरी में कैसे डायग्नोस करते हैं वायरस को, ये इसमें है। So first of all microscopy and stain. So we if we want to analyze the viruses, so electron microscopy is very very important for that, because the size is in nanometer. So for that we use electron microscopy. डायरेक्ट विजुअलाइजेशन होती है इसमें वायरसेस की। ज़ैंग स्मेयर इज वेरी वेरी इंपॉर्टेंट, एंड समटाइम दिस टी इज अ साइलेंट, सो ज़ैंग स्मेयर कहते हैं कई बार। फॉर एग्जांपल HCV हर्पेस सिम्पल वायरस और वेरिसोला जोस्टर वायरस में मल्टीन्यूक्लिएटेड जाइंट सेल दिखेंगे। इसमें क्या दिखेंगे, बहुत सारे न्यूक्लिएटेड वाले बड़े बड़े सेल। सॉरी। अनदर वी हैव अ नेगरी बॉडीज। नेगरी बॉडी सीन इन रेबीज वायरस, ब्रेन बायोप्सी करेंगे इसमें, उसमें हमको नेगरी बॉडी दिखेंगी गोल गोल सी स्मॉल अमाउंट में, राइट। अनदर टेक्निक वी हैव फॉर आइडेंटिफिकेशन लैबोरेटरीज कल्चर मेड मेथड. So we need not the artificial medium like nutrient agar, chocolate agar, like this thing. We need a living cell, right? There should be living being, because they are intracellular obligate. Obligate means necessary, and intracellular means inside the cell they need. Sorry. Then in this we have a three main thing: cell culture, egg inoculation, animal inoculation. In case cell culture, we used influenza viruses, then human herpes simplex virus, cytomegalovirus. For that we use a cell culture, right? In case of egg inoculation, influenza virus, this is very important nowadays also for vaccine formation, or for the growth, for identification and the research purposes. Animal inoculation, in case of rabies virus, nowadays this animal inoculation almost nil, right? But sometime we have to do. These two techniques are very popular: cell culture and egg inoculation. This cell culture also are primary cell culture, secondary and tertiary, so they are in different phases for different viruses, right? Another when we have a technique is a serology and molecular level. So in case serology, ELISA, either it to detect the antigen or antibody based on what we want to detect, like for HIV, hepatitis, and this is mainly a rapid kind of test mainly. Also we have a some other technique, but we have also some rapid kit for that. In case of serology, any part of maybe viruses, fungus or anything. Then we have a PCR, polymer chain reaction, detect DNA, RNA, highly sensitive. This is very important method in case of viruses, because they need some long time to show that their symptom to the body. In case of HIV, it may take 3 years to 10 years, right? So if we can't give a treatment, so it may be danger for the patient. So for that thing, it is very important if we have a small amount of DNA. So with this technique we can enlarge it and identify, yes, this is a HIV or another thing. Heme agglutination assay for immunoinfluenza viruses. Another we have a antiviral agent, so it may act against the viruses, this antiviral. So they have a different class: nucleoside, reverse transcriptase inhibitor, protease inhibitor. As the name suggests, they inhibit the thing, right? Neuraminidase inhibitor, interferon mainly boost the immune response. Neuraminidase block the virus release in case of influenza, viral protein in case of protease inhibitor, and reverse transcriptase inhibitor in case of inhibit reverse transcription. And न्यूक्लियस जो न्यूक्लियोसाइड है उसके अगेंस्ट क्या कैसे काम करता है, inhibit DNA polymerase. So these are the mechanism how these medicine act on the viruses to stop the infection spread inside the body, right? And these are the example on which viruses they act like that, right? This is also very important: prevention and vaccination. So we have a live attenuated vaccine, killed or inactive vaccine, subunit vaccine, mRNA vaccine. So these are the example: measles, mumps, rubella, varicella zoster virus, rotavirus, yellow fever, influenza, polio, SARS, hepatitis A, rabies, influenza, and for subunit hepatitis B, human papillomavirus. Then for mRNA vaccine, COVID-19, Pfizer and Moderna. So this is very important. We have some quick revision point: DNA virus replicate in the nucleus except the pox virus, RNA in cytoplasm except influenza, HIV, and virus released through budding, while naked virus through lysis. Segmented RNA virus, like you can name with pneumonic boar, bunny virus, orthomyxoarina, and Rio. Retroviruses, it have a reverse transcriptase enzyme. So as you see, we have antiviral category like a reverse transcriptase inhibitor. So HIV is our RNA virus. So from the RNA, it converted to DNA for their mechanism to cause infection inside the human body. But remember, if we have antiviral therapy which block this process, RNA to DNA, right? Or DNA to RNA in case of reverse transcriptase, DNA to RNA, because the HIV our DNA virus. So DNA to RNA, the process is blocked or inhibited by some medicine, So this is also a mechanism how the antiviral agent act, not for all but for the HIV, right? You can see there. So all these are the thing about the viruses. I hope this video is helpful for you. If you have any doubt, you can ask, and I hope the series is also helping you. Thank you for watching. We will meet in the next video.